Welcome to the Newsletter Out Loud. Our topic today is about total mixed rations and reheating. Maximize dairy production with FreshControl, keeping it cool, keeping it nutritious. 100 years ago, the feeding of dairy cows looked very different. Early guidelines for feeding concentrates were often based on a simple conversion, 1 kilogram of concentrate for every 2 kilograms of milk produced. Total mixed rations as we know them today did not exist and are, in fact, an invention of the latter half of the last century. A paradigm shift in dairy cow feeding. On the surface, the concept is simple: a method of feeding dairy cows that helps them achieve maximum performance. In a true total mixed ration, each mouthful of feed contains a consistent, definable, and as close as you can get it, nutritionally complete diet. Total mixed ration feeding allows cows to be grouped based on nutritional needs and can reduce the sorting of individual feeds by cows based on individual taste preferences. This results in a more stable and ideal environment for rumen microbes and ultimately improved feed efficiency. Silages often make up the largest portion of the various ingredients used to form total mixed rations. Silage, the foundation of total mixed rations. Silage has been used as feed for millennia. Good quality silage is highly palatable and nutritious and can be stored for a long period of time. However, silage quality depends on many factors, including crop maturity, moisture content, chop length, and temperature. In siling, the process of fermenting feed typically involves 4 main phases: aerobic, fermentation, stable, and feed-out. The aerobic phase occurs in the first 2 to 4 days. During this time, aerobic bacteria consume the oxygen in the feed, producing heat and carbon dioxide and creating an anaerobic environment. The fermentation phase takes place approximately 2 to 14 days after covering. During this phase, lactic acid and acetic acid are produced, resulting in a drop in pH. The storage phase begins at about 2 to 3 weeks. During this phase, the fermentation process slows down and the silage is stable. The pH remains low and the feed is protected from spoilage. The final phase is the feeding phase. At this point, the silage is exposed to oxygen. Microbial activity can increase, leading to a restart of the fermentation process and spoilage. The perils of reheating in total mixed ration. However, even using stable silage can cause unstable total mixed rations. The actual process of making total mixed rations exposes all feed components to air during mixing, which can trigger the reheating process. Sometimes, leftover total mixed ration from previous mixes can harbor high numbers of aerobic microbes. Even a small amount of residual total mixed ration added to a new total mixed ration can greatly reduce its aerobic stability and should therefore be avoided at all costs. The aerobic stability of total mixed rations varies greatly depending on the environment, temperature, formulation, and additional ingredients. Because of the frequently high starch and sugar content, total mixed rations can easily reheat at high ambient temperatures, resulting in nutrient and palatability losses. Researchers have reported that when incubated at 22 degrees Celsius, over 50% of total mixed ration reheats in less than 12 hours. This means that total mixed rations can spoil in the feed bunk even when fed twice a day. Some methods to reduce reheating include minimizing total mixed ration mixing time, maintaining mixer temperature, prompt total mixed ration feeding, and regular feeder cleaning. An additional method is to use preservatives, usually organic acids. These work by inhibiting microbial growth that can spoil the total mixed ration and reduce its nutritional value. Understanding the chemistry of organic acids. To understand how organic acids work, we need to explore the chemistry behind them. Organic acids occur naturally in plant and animal tissues. They can also be formed by microbial fermentation of carbohydrates, most commonly in the colon. They are a diverse group of compounds that contain a carboxyl group, which is responsible for their acidic properties. Organic acids are often used for their antimicrobial effects, which depends on their ability to dissociate in aqueous environments and lower the pH. This results in two main effects. The first is the effect on the environment in which the microbe exists. Most microorganisms have an optimal pH range for growth. If the pH of their environment falls below this range, growth and replication will cease. In fact, pH changes caused by organic acids are the mechanisms behind good silage. The second key principle about the effect of organic acids on microbes is that only the non-dissociated or non-ionized form of an organic acid can penetrate the walls of microorganisms and disrupt normal physiology. Once inside, organic acids dissociate into hydrogen ions and anions, disrupting biochemical processes. As the intracellular pH drops, the pathogen uses valuable energy to pump out hydrogen ions and restore the proper pH. Eventually, the pathogen cannot keep up with the energy demand, resulting in growth inhibition or even cell death. Organic acids can denature proteins, which are essential for many enzymes and structural components in cells. Because denatured proteins lose their natural shape, they are unable to perform their original functions. This can disrupt energy production, DNA replication, nutrient uptake, and other essential cellular activities. The negatively charged anions of organic acids can interact with the negatively charged phospholipid bilayers of bacterial cell membranes. This interaction can disrupt the membrane structure, leading to leakage of essential cellular components and compromising the integrity of the microbe. Combat microorganisms with salts of organic acids. Organic acids such as benzoic acid, sorbic acid, propionic acid, acetic acid, lactic acid, and their mixtures are widely used as preservatives and sanitizers in many industries. Some are more effective against bacteria, others against mold or yeast. However, the corrosive nature of pure acids limits their use. As a result, salts of these acids, for example sodium, potassium, or calcium salts, are widely used. Salts are not corrosive, which makes them easier to handle and use in equipment. Under acidic and humid conditions, salts of organic acids are converted to biologically active acids and have the same antimicrobial effects as the corresponding acid. For example, in the right environment, potassium sorbate loses its potassium, allowing the undissociated form of sorbic acid to passively diffuse through the cell membrane of pathogenic microorganisms. Inside the cell, sorbic acid dissociates and damages the cell membrane, inhibits enzymes, amino acid uptake, and RNA and DNA synthesis. As a result, the pathogen is unable to replicate. A similar pattern is seen with propionic acid, derived from calcium propionate. Propionic acid interferes with electrochemical gradients in the cell membrane, disrupts cellular transport processes, and inhibits the uptake of substrate molecules. This results in inhibition of microbe growth and preservation of feed nutrients and palatability. FreshControl, a powerful combination of organic acids. New EU legislation in April of 2023 reduced the maximum allowed level of sorbic acid in complete feed to take effect in early 2024. To meet these requirements, FreshControl has been reformulated to synergistically combine the preservative effects of two salts of organic acids to prevent microbial spoilage and nutrient loss in total mixed rations. FreshControl's combination of these two salts, potassium sorbate and calcium propionate, develops its antimicrobial effect only once mixed into the total mixed ration, when the two salts are converted into sorbic acid and propionic acid. Both acids have significant antifungal activity and complement each other by inhibiting the growth of bacteria and yeast. The newly formulated FreshControl was tested in 2 separate trials and has demonstrated its superior ability to control reheating and maintain aerobic stability. In one of these studies, 2 grams per kilogram of FreshControl was tested against a control for signs of reheating. Freshly prepared total mixed ration was placed into 2 4-liter buckets, one bucket containing only the total mixed ration, the control, and a second bucket contained the total mixed ration mixed with 2 grams per kilogram of FreshControl. Both buckets were placed outdoors in the shade at ambient temperatures, an average of 23.8 degrees Celsius, for 30 hours. Each bucket was divided into 4 quarters where the hourly temperatures were measured, and the results averaged. Throughout the test period, and despite a sharp increase in ambient temperature, 2 grams of kilogram FreshControl added to fresh total mixed ration prevented reheating. FreshControl effectively tackles the challenges of aerobic stability in total mixed rations. Don't let spoilage compromise your total mixed rations quality. Say goodbye to spoilage and nutrient loss and embrace the freshness and stability that FreshControl delivers. Keeping it cool with FreshControl. I'm Dr. Melinda Culver for Biochem, and thank you for listening to the newsletter out loud. If you would like to learn more about this topic, please visit our website at biochem.net.